going to talk about specificity, okay? So what was the meaning of affinity? What's the other word for affinity if you want? Say it loud. Strength, okay? Affinity is strength of binding. What variable do we use to measure it, if you will? KD. And what units does it have? And the lower the KD? Great. All right, so real life, real systems don't have one-to-one interactions, okay? In reality, we'll have ligands that have many possible binding partners. So a given ligand can bind to different proteins, different enzymes. It can bind to nucleic acid, whatever it is. And the relative probability of forming the different complexes will determine the functional outcome, right? So when the ligand binds to the protein, when the ligand binds to a different protein, different things might happen, right? Okay? And so sort of relative probability is going to come into play once we put the mathematical relationships to this. Okay? So what do you think controls sort of your relative likelihood of binding to one protein versus a different protein if you're a ligand? What is the word we just defined 5 seconds ago? Affinity, thanks. High affinity. The higher the affinity, the better the binding, except that high affinity is insufficient to ensure that you'll have sort of biologically relevant activity of these ligands, because if stuff is really, really high affinity, it's really, really strong. Another way to think of this is it's really, really sticky, but this ligand then just sticks to everything, and there's a whole bunch of off-target binding that you do not want, okay? In order to have high affinity interactions work within a cell, they also have to be very specific. So you need high affinity for your target receptor and low affinity for all of the other receptors. Okay? So we're looking for a KD that's really tiny for the receptor of interest and high KDs for everything else. So specificity can be expressed mathematically in terms of the affinity of a ligand for its particular target relative to all the other potential targets. Like I said, it's a relative probability, so maybe not so surprisingly, this is about to look like target of interest on the top, the sum over all the other possible targets on the bottom. So this is we're building to. And this is just something of importance to note. Specificity can be thought of in sort of a reverse way, rather than saying, I'm a ligand and I can bind to all these different receptors. You can just be looking at one receptor and thinking about all the different ligands that could bind to it and still calculate specificity. And so this is, again, going to be one where you are asked to read carefully what you're being asked to compare, okay? So again, it could be relative strength of one ligand versus other ligands to a single receptor, or one ligand across multiple receptors. So fibroblast growth factors, or FGFs, are a family of proteins. This is just an example that we'll use to calculate specificity. FGFs are a family of proteins that primarily regulate angiogenesis. They also have, depending on which FGFs you're looking at, they have involvement in wound healing and laying down new collagen. Okay. There are 18 known FGFs in humans, and 4 genes, each with 3 different splice variants that code for FGF receptors, making a total of 126 possible combinations of different FGFs to different FGF receptors. Okay? And so since any FGF theoretically could bind to any one of these receptors, there's sort of a lot of this possible off-target binding that you have to consider. FGFs, however, happen to be really selective. Okay? So there's a couple different variants of the FGF molecule itself, 1 through 6. And this is a sort of a graphic from your book showing these are the different receptors. Okay, receptor 1, 2C, 2B, whatever. And so it shows strong interactions or high affinity being thicker lines, weak interactions being thinner lines. And if there's no line connecting those two, it's effectively absent. You don't even have to consider that as a possible off-target site. Okay. Selectivity is obviously really important so that the right function occurs. And so this was maybe an example of what are the negative consequences of like downstream. So FGF can be involved in angiogenesis or making new blood vessels. They can also be involved in wound repair. You don't really want to be synthesizing connective tissue when you're supposed to be making a blood vessel. And so this is an example of, oh, hey, am I laying down like scar matrix? When I'm supposed to be making a blood vessel, it's not going to have the flexibility it needs. I'm not actually going to be able to pump blood through that properly. So this is an example of if you had sort of some miscommunication of FGF and its receptor, what sort of downstream effects you could have. Okay. Cells generally will only express a few isoforms of the receptor, so you're not really worried about all of the possible combinations because cells aren't actually making all of them. And you can have some sort of post-modification. So who remembers what glycosylation was from way back? Just broadly, like what do you think it was from chapter, I don't know, 3? Great. Adding a sugar to a protein. So you can glycosylate a protein. You could glycosylate like the surface membrane of a cell surface to the lipids. You could add glycosylation to anything. But yes, it was adding these sugar molecules, these glycan molecules, to sort of modify what's going on. And so if you have sort of a specific pattern, then you'll have specific binding between these two things. This is a table from your textbook. Okay, it's giving this what this table is showing. We will do an example with it. I will almost 100% ask you to use it on either the homework or exam. Okay, what's in here are KDs. Okay, these are dissociation constants. And what we have are dissociation constants between different FGF ligands, and then here are the different receptors. Okay? And so again, this is an example where I could ask you to determine the specificity for FGF1, for FGF1 receptor, let's say. This is its target, and you need to sum over its interactions with all the receptors. Or you could be asked, what is the specificity for, say, FGF1 for F2 versus all the other ligands? And in that case, here's the target. Here's all the other ones you're summing over. And again, the language of the question will help you know this. If you do it backwards, it's like almost none of the points. So, okay, N sub D, or N sub D, there's no sub, ND here, this means not detectable. And the instructions, I've cut the caption off of the bottom of this, but sort of the instructions in any problem you'll be given is that not detectable, you should go ahead and let K sub D be 10 to the negative 4th. Okay? And it's basically just, that's a really high value for KD. We said that stuff that's high affinity is down in like the nano and picomolar. So this 10 to the negative 4th is just, it's not a strong enough interaction to be worth considering. It's very high.